Welcome everybody. We will now resume our network analysis workshop, and I'm very happy that Gary Bader from the University of Toronto is willing to show us some of his exciting pathway tools. You remember that Gary is one of the people who helped build the new functionalities for Cytoscape, but he also did a lot of research on pathway enrichment analysis tools. So with that, please start.
Thank you very much, Steve. So I'm going to cover, I have an hour and a half to cover material here. So I'm going to cover first some methods that we find very useful for pathway analysis of genomics data. And then depending on how much time we have, I can go into some analysis later on interpreting mutations from cancer genomics data sets. And so my lab is really focused, I guess, my research is really focused on trying to collect as much pathway information and cell mechanism information as we can get our hands on. And the idea is that with genomics data, we get sort of information about the state of the system, the cell, or the tissue at particular time points or under specific conditions. And we want to combine that with all this mechanistic information to get some mechanistic understanding of how that state of the cell came about. For instance, was there a particular transcription factor or microRNA that was important for activating the pathways that you see changing in that particular condition? So just to give you an example to start off about pathway analysis the type that I've done is this picture from a paper that we had from a few years ago in collaboration with Stephen Scherer's lab at the University of Toronto at the Hospital for Sick Children. He studies autism spectrum disorder. And they were, I'll talk more in detail about this example later, but just to give you a quick summary, they were interested in copy number variants, and in particular rare copy number variants that were associated with children who had severe autism spectrum disorder versus controls. And when they looked at individual genes, they didn't find very many genes that were significant after multiple testing correction. They had maybe 3 or 4. And so when we wanted to look to see if there were any pathways associated with the autism spectrum disorder, and we found a rich set of pathways that were quite strongly associated with the spectrum disorder. So and the interesting thing about this was that a lot of these pathways gave some information about the mechanism that could be explaining the pathology, so GTPase and RAS signaling, for instance, kinase activity, some pathways that are involved in neural differentiation and branching of neurons. And the interesting thing was that when we looked at these individual pathways, we didn't see the same mutation over and over again in any of the patients. There was about 1,000 patients. Instead, we saw the same pathway, but different genes mutated. So when we looked at an individual pathway, the signal was coming from different genes. Each one was mutated in 1 or 2 samples. But when you looked at the pathway together, that pathway was mutated in many samples. So that sort of increased the statistical power of the approach by collecting information from multiple genes based on our knowledge that they're involved in the same pathway. So that's kind of an example. And the idea I'm thinking that I'm promoting more is trying to go, in this case, this was like a genome-wide association study where you're looking for mutations or genetic markers that are associated with a phenotype. And that's a correlative approach where we find that a particular disease gene is correlated with a phenotype of interest, or a particular gene. And it's a very powerful approach. It's been used over many decades to find many of the disease genes that we know about. But there's a couple of problems with it that I think Steve mentioned yesterday, that the more markers you look at, the more SNPs you look at, the more rare, the more copy number mutations you look at, the more multiple testing correction reduces your statistical power. And so pathways reduce the number of variables that you're looking at, and thus that helps with that and increases statistical power. And it's also more intuitive to understand if a pathway is changing versus an individual SNP, for instance, is changing, or a copy number variant, because we can read the name of the pathway and it tells us something about the biology. And so those are kind of two useful things about pathway analysis. It can help increase the statistical power, and also it helps with interpreting the results. Okay, so just another way of thinking about some of the methods that I'll talk to you about is that they're useful for interpreting gene lists. And in the case that I mentioned with autism spectrum disorder, the gene lists were genes that were affected by gains and deletions in a copy number analysis. But those gene lists can come from anywhere. They can come from differentially expressed genes. They can come from proteomics experiments. Any large-scale genomics or omics technology that generates lots of genes we can apply the methods that I'll talk to you about on. So and I think everyone here knows this already. It's the theme of the workshop. When you've started doing some genomics analysis and you are happy that it worked and you get data, but then you realize that there's thousands of data points, and now what do you do, right? So, and often people want to know if you have a list of genes, people want to know what's interesting about those genes. And a very popular method, very useful and frequently used method to search for interesting things about genes is to find out if they're enriched in known pathways, complexes, and functions. And so I'll talk to you more about this. And in general, I like to think of pathway and network analysis as any type of analysis that involves pathway or network information, which a number of examples in this workshop there's a lot of different examples of types of analysis that are presented in this workshop. We most commonly use it to interpret lists of genes. The most popular type is pathway enrichment analysis. There's tens of thousands of papers that have used gene ontology enrichment analysis or some type of pathway enrichment analysis. And as I said, this type of analysis helps gain mechanistic insight into genomics data and hopefully get at a little bit more causal insight, so you can understand what's causing the state of the cell that you're looking at. Okay, so I'm going to now focus more and talk about pathway enrichment analysis. And the general idea, I think I mentioned this yesterday for people who were in the workshop, is you are taking a list of genes and a list of pathways, and you're trying to find out if any of the pathways are enriched in this gene compared to what you'd expect given all the genes in the genome. So here's a couple of pathways involved in spindle, say, assembly and apoptosis, and there may be genes here that, if half the genes in this list are involved in apoptosis, that's many more than you would expect would occur if you just randomly selected genes in the genome. Then that's enriched, and you can assess the p-value of that and get a score for how enriched that is. And a lot of, how many people have used this type of enrichment analysis already? Okay, so quite a few. So those people, who've used it, you probably have seen the result, which is usually a big list of pathways and their scores. So how significant the enrichment is in the list of genes. And it's an excellent idea. As I mentioned, it's been used in thousands of papers. If you look at David or GSEA, they've been cited, if you collect them all together, more than 10,000 times. But one of the problems with working with this type of information is that there's a lot of redundancy in these lists. So for instance, you can see immune effector process and myeloid cell differentiation. If you're a biologist, you might recognize that those are both related to the immune system. If you don't know, all of the terms, then you might not recognize that. But it's important to recognize that those are related, and they probably have a lot of genes in common. And so finding sort of the major themes, the major pathways or processes that are affected in the experiment can be sometimes problematic or more difficult when you are faced with these big lists. So there's this, there's a lot of overlap, and there's a burden in trying to figure out all this overlap. So a postdoc in my lab, Daniela Marico, and Ruth Isserlin, a research assistant, developed a method. We developed a method to visualize the results of pathway enrichment analysis. And this is useful anytime, for any kind of enrichment analysis that you've done. So if you are looking at enrichment of specific gene ontology terms or pathways that you've gotten, or molecular signatures or disease-associated genes, your user-defined gene lists to see if they're enriched, all of those things can be input into the software to visualize how the gene sets or pathways are related to each other. So I'll explain this in a little bit more detail in a few slides. But the basic idea is that we take the pathways that are enriched, and I'm using the term pathway enrichment analysis, which I think is more understandable to most biologists. Pathways are represented as gene sets of genes, and those sets of genes can be things other than pathways, but I'll just use the word pathway. So there's a set of pathways, and you have found out which ones are enriched, and then you visualize it using what we call an enrichment map. And the enrichment map shows each pathway as a circle, and it relates them based on their overlap. And you can see that there's a lot of related pathways here that are all part of one big theme, in this case, DNA metabolism, cell cycle. I'll go into more detail about this. Okay, so in the next few examples, I'm going to be using a pathway enrichment analysis method called GSEA, Gene Set Enrichment Analysis, which is developed by Jill Messerov's lab at the Broad Institute. And in GSEA, it's often used for doing pathway enrichment analysis on differentially expressed genes from microarray or RNA-seq gene expression data. But it can be used for any kind of ranked list where you have a list of genes that are ranked by some measure. So this is the list. These are and so the examples that I'll show are all related to gene expression data. And so in this case, the genes at the top of the list are all going up in your experiment versus your control, and the genes at the bottom of the list are all going down in experiment versus control. Then GSEA is used to identify pathways from the database that are enriched in the top of the list or enriched in the bottom of the list. And these are colored red for genes pathways that are in the top of the list, and blue for pathways in the bottom of the list. And we interpret this as sort of pathways that are going up, and blue as pathways that are going down. Okay, so again, we start with this list of ranked genes. We run our GSEA or other enrichment analysis to find pathways that are enriched, and then the enrichment map comes in. As I said, each circle here in the map is a pathway. The size of the circle is proportional to the number of genes in the pathway, and the connections, the edges that link the nodes are weighted based on the amount of overlap between the pathways. So this can be, you can think of this as pathway crosstalk. Basically, an edge means that the pathways have some number of genes in common. And then we color the nodes based on their enrichment score. In this case, because GSEA gives us things that are enriched going up and things that are enriched going down, we have the intensity of red means a more intense red color indicates stronger enrichment in pathways that are going up, and similarly for blue for going down. Okay, so Enrichment Map is useful right now for 3 major types of analysis. We call them use cases. The basic one is a single enrichment analysis. So this is, you've done your gene ontology enrichment or your pathway enrichment on a particular data set that you have, a particular gene list, and you want to visualize the results as an enrichment map. So I'm going to be using a data set that was published. We found it in the Gene Expression Omnibus database. This is a the researchers who produced this data set were studying breast cancer cell lines, and they were interested to see how genes were changing when these cell lines were treated with estrogen. And they looked at multiple time points. They had 3 samples for estrogen treated and 3 samples for estrogen untreated at different time points. So we're just focusing right now on the 24-hour time point, on one of the time points. And we ran our enrichment analysis with GSEA, and then we visualized the results as an enrichment map. So the enrichment map, I'll tell you how to make these enrichment maps using Cytoscape in a bit, but the idea here is that, what was quite interesting when we first looked at these enrichment maps is how much redundancy and clustering there was among pathways. So for instance, all these little red circles here are different pathways, but they're all related to ubiquitin-dependent protein degradation. So imagine these pathways, 10, 15 pathways here, are spread out over a whole list, and they all have different names. You can sort of see here, actually zooming in on this microtubule cytoskeleton one, there's all sorts of different names here: centrosome, spindle pole, microtubule-based movement, but they're all related. In the enrichment map, you can see how they're related, and you can see that there's a general functional theme here, with and we label them with some general label. So this gives you a very nice kind of bird's eye view, or a nice summary of your genomics data in terms of known pathways. And again, the red means going up and blue means going down. So right away I can see that a lot of pathways are going up and a few pathways are going down, and I can zoom in on some pathway that I'm interested in. Maybe some of these pathways are already well known to you based on your expertise in studying the system that you're looking at, and so those might not be as interesting. And others you might not have seen before, and so maybe you want to zoom in on those. So you can kind of, as I mentioned, zoom in and see the individual pathway names, and you can zoom in further, which I'll tell you about later. Okay, so the second workflow or use case is a comparison of 2 enrichments. So in this gene expression study, the investigators looked at multiple time points. So we did an enrichment at an early time point and a late time point, and they were interested to see we were interested to see how pathway enrichment is changing between these 2 time points. So we made enrichment maps of each enrichment result at each time point, and we put them together into one map. And this is easy to do with the software. And in this case now, instead of having one color for the node, we have 2 colors. The node center is representing the enrichment at the early time point, and the node border is, the color of the node border represents the enrichment score at the late time point. And so we can see, for instance, here's an example where there's, actually let me find this. This example has a red border and a white center. So white means it wasn't enriched at the early time point, and the red border means it was enriched at the late time point. So we can see a lot of nodes or a lot of these circles are all red, which means that there was, the pathways were enriched at both early and late time points. So a lot of pathways are not really changing between the early and late time points. But we can find a few areas where things are changing. For instance, in this portion of the map, there's quite a lot of change. So ubiquitin-dependent protein degradation, cell cycle, and microtubule cytoskeleton generally weren't as enriched at the early time point, but they're much more enriched at the late time point. And the reverse is seen for this little part of DNA metabolism here. So this is very useful because if you didn't have this visualization tool, you'd be looking through big lists of pathways and trying to figure out which ones were mapped to each one. And some people might be in the audience, I see, might be nodding that they've seen this. So so this visualization technique really just helps you quickly zoom in on things that are different. And in the software, you can click on if you've loaded up gene expression data, again, I'm using that as my main example, you can click on each circle, which represents a pathway, and see the genes in that pathway and see how they're different, the actual gene expression data. So here you can see that these pathways are kind of more active compared to control in the late time point, and you can see that there's not much change in the early time point, and in the late time point there's a really big change. Similarly here in the replication fork, there's a big change at the early time point, and at the late time point, they're both similar. It's important to realize that they might be up or down, but we're looking for change between the treated and untreated, or the experiment and the control. Okay, any questions so far? Yep?
Yes, the labels that you have on each big circle, are they generated automatically, or are they human curated?
So the question was, are these labels generated automatically or manually? So currently they're manual. We're looking for a way of making them automatic. I'll mention that in a bit in future directions. But when you generate an enrichment map, it just shows you all the circles and lines. It doesn't show you the bubbles and the labels. And so we do that manually, and we hope to automate that in the future. Okay, so the third thing that is useful in Enrichment Map, the sort of last thing that we use it for, is what we call query set analysis. So there's a number of different biological questions that you can answer with this, but the idea is that once you've done your pathway analysis and you've identified a bunch of pathways that are going up and down in your gene expression data, you might have another list of genes that you're interested in to see how those genes relate to the pathways. So for instance, you might have genes that are associated with your disease that you're studying, or you might have genes that are targets of a microRNA that you're interested in, or a transcription factor. And then you can see how that set of genes relates to the pathways. Is there overlap between your gene set of interest and the pathways that you've seen enriched? So here we took some gene expression data from a mouse study where they knocked out a particular microRNA in mouse heart tissue, and they collected gene expression data before and after. They compared the knockout versus wild type, and all of these pathways here are enriched in all the pathways that are red are enriched in the knockout condition, and there's a few pathways that are going down. So this kind of makes sense because a microRNA is a negative regulator of gene expression of its target genes, and so it's nice to see that a lot of pathways are going up, because when you remove a negative regulator, you expect a lot of things to go up. Some things are going down as well. That might be indirect effects. So then we took the predicted targets of that microRNA from a target scan database, and we represented that set of targets as another node. In this case, it's a triangle here. And then we computed, we used this query set analysis to just compute the overlap between these sets, all the sets. And only the significant overlaps are shown, and you can choose the sort of significance level that you want to see. But again, it's interesting to see that some of these pathways that are going up seem to have a lot of genes that are known or predicted targets of this microRNA. And some of these pathways have a lot more microRNA targets than others. So for instance, vesicle trafficking has a lot of genes that are a lot of microRNA MER1 targets, MER1-2 targets, and some other things like this mitochondrion theme here doesn't have any targets in it. So this kind of argues that some of these pathways might be directly targeted by the microRNA and others are indirectly targeted. It's nice to see that none of the pathways that are going down are directly targeted by the microRNA as validation of what we'd expect. So again, just zooming in here, the enrichment map shows the pathways, the pathway activity in the experiment, and then the query set is an additional set of genes that you can add and look for overlap with those pathways. Okay, so we published this method a few years ago, and we've been using it all the time in our lab and we're trying out different ways of using this method, this visualization method. And so now we have a few papers that are out that have used the method in different ways, and I include just some citations here. So and I'll just mention a few of them. So this so you can get a sense of how it's useful. So this is sort of just a normal enrichment map. In this case, it was looking at a breast cancer tumor initiating cell, and the senior investigators were interested just to see which pathways were characterizing different cell type populations that they'd purified from breast cancer. So this helped them make a stronger conclusion that their cell types that they were purifying were very different from each other, because they exhibited very different types of pathway activity. They were looking for stem cells. Here's another couple of examples that use this query set analysis. So in this example, investigators John Dick in Toronto was looking at gene expression in hematopoietic stem cells that where they already knew that a particular microRNA was important. And so this query set analysis kind of helped, we first showed pathway activity, and then the query set analysis helped kind of prove that this microRNA might be actually regulating some of these pathways, or maybe not prove, but suggest. And then they followed that up with additional experiments. This is a similar example with a transcription factor. So here, the gene expression data was, again, showing which pathways were changing. And then we looked for transcription factors that were very differentially expressed. We used the opossum tool from Wyeth Wasserman's lab in University of British Columbia, which helps rank transcription factors, the activity of transcription factors in gene expression data. And HIF1 alpha came up as the sort of top transcription factor. So we looked at the targets, predicted targets of that, and saw how they connected to certain pathways. And this provides a somewhat of a mechanistic hypothesis that HIF1 alpha might be an important transcription factor in the system that they were looking at. So I forgot to mention one thing earlier, and I'm mentioning hypotheses. The power of pathway enrichment analysis and the visualization method that I'm talking about is really to help sort through a lot of data and identify certain hypotheses that you might want to follow up on. So you kind of want to get from a lot of data to some specific hypothesis as quickly as you can. Although sometimes you can get some very specific mechanistic hypotheses. Okay, so I'm going to go now back into the autism spectrum disorder project and talk about it in a little bit more detail because it kind of uses all of the different aspects of this enrichment map that I'm talking about. So when I got involved in this project, I was interested to learn that autism spectrum disorder is highly heritable. It has up to 90% concordance in twin studies. I didn't realize that. It's not always severe in each child, but sometimes it's very mild and sometimes severe. But there's also a lot of known, some of the genetics of the disease have been explained by rare single mutations. But when the study was published just before, they were increasing evidence to suggest that de novo rare copy number variants, these are copy number variants that were not present in the parents, and they were kind of generated de novo in the child. They might be they might be involved in autism spectrum disorder. So Steve Scherer and colleagues collected about 900 cases and over 1,000 controls, and they used the Infinium 1 million SNP chip at the time, that was state of the art, to measure intensity of DNA at different SNP positions, and then they used that to predict copy number variants. And then they were interested in just the rare copy number variants from their hypothesis that the rare variants are more important. So they only looked at variants that were found in less than 1% of the population. And they looked at the individual genes, as I mentioned, and then we ran pathway analysis, pathway enrichment analysis that I'll tell you more about. It's slightly different than the GSEA analysis. And we visualized this as an enrichment map. And the circles represent the pathways that were associated with autism. And to convince, there's a lot of new pathways in this result. And some of the pathways they'd never seen before. And some of them are fairly general. So there's an annotation problem here because some of these pathways are probably involved in neural processes, but the gene ontology or the pathway databases have not, at the time especially, had not annotated a lot of neural related pathways. So although there were some, like cell projection and cell motility, and some of these pathways are related to neural development. This right here in particular central nervous system development. So, so to convince our colleagues that these pathways were interesting, we looked at genes that had previously been associated with autism and also intellectual disability. And they had collected more than 100 genes that had been associated to these phenotypes. And we did pathway analysis on those as well. And so this triangle represents intellectual disability genes, and these triangles here represent pathways that are enriched in the known intellectual disability genes. Similarly, for autism, there's a set of pathways that are enriched. And even though there weren't that many genes, weren't really genes in common between the ones that were affected by copy number variants and the ones that had previously been seen by, for instance, genome-wide association studies, they were both enriched in pathways that were similar. So autism genes were enriched in pathways related to behavior that also came up in the copy number variant data. Intellectual disability, there was a lot of pathways involved in central nervous system development that were enriched, similar to the copy number variant data. Even though the genes weren't found to overlap hardly at all, the pathways definitely overlapped in a rich way. And you can kind of zoom in here to see this in a little bit more detail in this CNS development cluster. So this just shows you a little bit more detail here. Okay, so the statistic that we used to compute this was a little different than the typical enrichment analysis that people do using Fisher's exact test or hypergeometric distribution, type tests, or the GSEA ranked analysis. We were interested in developing we actually tried those methods, and they didn't give us very many hits. The reason why is that this copy number variant data, remember, they were really focusing on rare mutations. So if you had a gene and if you ranked genes by the number of patients that they were mutated in, it wasn't very many mutations. It was never more than a few patients for an individual gene. That was the frequency of mutation. So if you rank those, you just get a lot of ones, twos, and threes. It's very difficult to rank hundreds of genes that way. So there wasn't a lot of information there for ranking. So we tried a self-contained, what some people call a self-contained statistical test for instead of doing a gene association, we did a pathway association. So the way this worked, it's very simple. Each green box here represents a pathway, and each little box inside represents the genes in the pathway. So there's a bunch of genes in this pathway, a bunch of genes. So this box represents one pathway. Each column here represents a different patient. And then we look to see in an individual patient if the genes in that pathway were mutated. And each time a pathway was mutated in one patient, it would get a count of 1. So even if there was many more, so if there was one gene mutated in the pathway in a patient, you get a count of 1. If there's 3 genes, you get a count of 1. If there's 2 genes, you get a count of 1. And you get a count of 0 only if there's no gene. So basically, we're looking for pathways that were affected in any way or not affected in any way. And then we collected all that information, and we looked at the number of patients where a pathway was affected in cases versus controls. And we ran a standard Fisher's exact test, which is the same type of statistics used for genome-wide association studies. And we assessed the false discovery rate by shuffling the columns in this matrix thousands of times and seeing how often we get this type of a particular enrichment for a particular pathway by chance. And so just to quickly summarize that, in this case, we mapped our mutations to genes. We generated this big matrix of pathways versus patients. And then we used the counts for a standard Fisher's exact test, standard association test. The pathway databases that we used were mostly gene ontology and a few pathway databases at the time, KEGG, NCI, and Reactome. And we also used PFAM domains, although those didn't really come up in the result. We filtered our pathways. So if we just took all of these pathways together, we'd have 14,000 pathways, which is a lot. we only looked at pathways that were that contained between 500 and 700 genes. If you have pathways that only have 1 or 2 genes or 3 genes, it's often difficult to kind of get a good statistic, a good estimate of how likely those pathways are going to be enriched by chance. And if you have pathways that have hundreds of genes, usually those pathways are so general that when you get them back, you don't, they're not useful. So they might be something so general that you don't want to see them. So we just remove them. And by removing them, we reduce we cut our number of pathways by half, and that helps with statistics. And also, in the end, there was only 3,500 pathways that had some mutation associated with them. Because there were, this was a new data set at the time, copy number variants, so we'd never run this type of analysis. We ran some additional tests just to check for biases. So copy number mutations can be really short or really long. We removed the super long ones that are whole chromosome or half chromosome, big chunks of the chromosome, but there's still potentially a length bias. And people know, for instance, that sometimes if you have a cluster of genes in the genome and that are functionally related, like olfactory receptors, which all cluster together, and if you have one mutation that affects all of olfactory receptors, it will affect a lot of olfactory receptor genes, and you'll get a really strong olfactory pathway coming up. We didn't have that issue here because of this counting only one patient per pathway here, but there may have been some bias in length. So we tested that, and there wasn't. There didn't seem to be any relationship between how confident we were that about our enrichment as measured by false discovery rate versus the length of copy number variants. We also looked to see if genome proximity among genes in a pathway would be somehow biasing our results, and we didn't see that either using a similar analysis. We did see that all of the enrichment was coming from deletion genes, and there was not really any pathway enrichment coming from gained mutations, so it seemed like it's really all about deleted regions of the genome in this disorder. And then we looked further at the list of autism-implicated genes and intellectual disability genes, and we looked at these individual themes here, these what I call functional themes, for instance, cell motility or GTPase and RAS signaling, and we tried to look more specifically to see if the genes involved in these systems were more often found in these known disease genes than we would expect. And in general, the certain themes that we focused in on were found more often in known disease genes. So that sort of, again, tried to validate the theme. So I went through that in a little bit more detail just to give you an example of some of the additional analysis that some people try to do on top of pathway enrichment analysis to convince themselves of the results. And that was actually more than we typically do, but it's interesting to see. And we tend to do that more if it's a new type of data that we haven't used before, like copy number variants. But if it's gene expression, we're very where we've done it many times, we won't go into as many side tests. Okay, so Enrichment Map is I have a couple more examples to show you, but I just wanted to talk about the software itself. So Enrichment Map is freely available software implemented as a Cytoscape plugin. So we learned, people who are in the workshop learned about Cytoscape yesterday. Cytoscape is free software for network visualization and analysis for the people that weren't here yesterday. And this Enrichment Map plugin can be used to load up enrichment analysis and I don't list it here, but we actually support David, GSEA, gProfiler, and generic formats that can be just generated very easily from our code or anything like that. So any kind of enrichment analysis method can be used to generate results that can be loaded up into the system. If you have gene expression data, you can visualize it. As I mentioned, you can click on these pathways and see the genes in the pathway. We have a little bit of extra work that we've done to coordinate with the GSEA team. And actually the next version of, how many people here use GSEA, gene set enrichment analysis? Okay, so a handful. One of the next versions of GSEA will actually have Enrichment Map built into it. So from your GSEA results, you'll just be able to click Visualize, and you'll get one of these maps. For people that are familiar with GSEA, there's an interesting aspect of GSEA that tells you the genes that are most contributing to the enrichment signal, and they call it the leading edge. And so in this case, we highlight those here, just as it's a nice feature for GSEA users. Okay, any questions? The enrichment map is available now. The GSEA integration is probably coming out in a few months, I would think. And if you want to try an early version, just email me, because it's ready to use. It just, they have to release it in one of their next release cycles. Yeah? Sorry, some genes may be.
Some genes maybe have
multiple, yes, okay.
Multiple gene mutation, multiple co-terms. So co-enrichment may have some overlapping before. Yes. So how does the pipeline handle that overlap?
So there's two questions. The first question is about the imbalance of gene ontology terms in terms of the number of genes that are annotated to each term. So some genes have only a few, some Go terms only have a few genes, some terms have many genes. The second question is that there's an individual gene can be annotated by multiple Go terms, and so there's overlap in the Go terms, right? So the first, the statistical tests that are used for enrichment analysis, the standard ones that most people use don't consider those. There are a number, in fact, it's an active research area in statistics to develop new methods, new enrichment methods that do consider overlap between terms. If terms are overlapping in terms of the number of genes that they annotate, then they're not independent, right? And so if you do multiple testing correction, it assumes independence between the tests. And that's not the case, really. So multiple testing is not going to be accurately, that's one statistical problem, is that multiple testing is not going to be accurately assessed by the standard methods. So there's a whole bunch of statistics methods that are trying to address that in the enrichment test itself. Those have not really reached the mainstream, although there are many tools available. Most people use these standard tools. The questions that you had are really one of the reasons that motivated us to make this enrichment map software, because we get to visualize the overlap and we visualize the number of genes that are associated to the Go term. So the size of the node is proportional to the number of genes associated with the Go term. So you can see the imbalance if you want to see the different size nodes. Similarly, Go terms have genes in common, and all of the Go terms that have a lot of genes in common are grouped together in this analysis. So again, you can see the redundancy. We don't correct for that redundancy in any way currently. This method is just visualizing the results. So it's, I should say, we don't correct in any automated way. We, when you visualize these results, you can immediately see those problems if they're problems, and then you can interpret this map thinking about those. So I think it's similar in approach to what Steve and others have been talking about in this workshop, that it's really important to visualize the data, right? To see all the data, and this is a method for doing that. But it doesn't address your concerns in terms of the statistics. One thing that's useful is that it provides these slider bars that allow you to interactively change the threshold, your p-value or your q-value, the significance scores for the terms. And as you slide these along, you might see certain themes just disappearing very quickly. And so that means that the whole theme, including all the terms that are redundant, is not very robust. And other themes which might have 20 pathways all as part of one theme, some part of them is disappearing when you increase the threshold, make the threshold more stringent, but there's a core that's always there, and it's even there when you increase the threshold to its most stringent level. And so that can help you kind of identify the confidence in a particular theme. One of what I call these clusters here, or these modules, I call them functional themes. So again, it's an interactive visualization response to your question, but it doesn't automatically figure out the result. Does that answer your question? Okay, any other questions? Okay, so I'll continue talking about a few more things with Enrichment Map, but one of the questions earlier was, about, well, I guess one of the questions earlier was about automatic labeling. So as I mentioned, and I'll just skip to that, as I mentioned, the Enrichment Map you get from Cytoscape, from the Enrichment Map tool, is just shows you all this network by itself. And the publication quality enrichment maps that I showed had bubbles drawn around the modules and labels. So we have a tool to help you label things. It's a word cloud plugin that you can select a set of pathways, and then it will tell you the frequency of words in those pathways. And so this region here, this enrichment map, has a lot of signaling pathways in it. And so you might, and it's actually a few different types. So this kind of helps you identify labels. We also are thinking about this in a general framework of, as I mentioned, the Enrichment Map kind of gives you a summary view of your genomics data. Then you want to zoom in to an interesting, you want to identify an interesting region and to follow up on, right? And so that interesting region might generate some hypotheses that you want to follow up on. But you also want to get more mechanistic insight into the genes in that region. And so we've manually done this, and we want to automate it. We want to have it automatically part of this software to identify a region. In this case, we have an enrichment map, and we found that apoptosis was really changing, and it also looked interesting because of expertise in the area. So we zoom in on that one. And then we take one of the pathways here, which is from Reactome pathway database, and we now switch to a view where instead of nodes being pathways, nodes are proteins, and lines are protein interactions. And so in this case, we're actually looking at the apoptosis pathway as a pathway diagram, and we overlay the gene expression data on it, and we can see there might be some part of this pathway that's really differentially expressed. And so that now gives us and we can zoom in on that. So in this case, this was protein expression data actually, and we followed this, and it led us to this caspase set of complexes that's important in apoptosis that seem to be really important in this study. So that is one of the future directions we want to kind of help people get down to a more mechanistic understanding of any genomics data set. Okay, so one last pathway enrichment story that I really like. Then I'll move on to other topics. Some general summary, and then move on to some other topics. This is a collaboration with a neurosurgery lab in a research lab in Toronto, who studies, one of the things they study is they study rare brain cancer in pediatric populations, medulloblastoma and ependymoma. Again, in collaborating with Michael Taylor, who's the surgeon and who runs the lab, I learned about a little bit some interesting things about these disorders. So ependymoma is a brain cancer that affects the lining of the brain, the ependymum, and it's the among ependymomas, the most common and morbid location for children is in the posterior fossa, which the Allen brain atlas people will know better than me, but from what I learned is the brainstem plus the cerebellum. So Michael Taylor had previously looked at gene expression data and found that there was two major classes of ependymoma. Class A, which primarily affected the youngest patients and had the worst prognosis, and Class B, which primarily affected older patients and generally had a very good prognosis. So gene expression clustering identified these, and that's been very useful for their lab. They wanted to investigate this classification further. So in collaboration with a German group, they did exome sequencing of 30 samples, and they did whole genome sequencing of another half a dozen samples. And interestingly, there were no recurrent mutations at all, and almost no mutations at all in general. There was only a few mutations, like 2 or 3 per sample. And it's not totally surprising given it's a pediatric population. People in cancer genomics have found that, mutations build up over time, and so the younger the sample, the less mutations you expect them to have, expect the sample to show. So they tried something else. They looked at methylation arrays, and on 79 samples, this is DNA methylation, and they found that there was actually, this showed the same clear clustering into A and B classes, where the A sample was more transcriptionally silenced by CpG methylation. And that A sample is the more serious one with the more dismal prognosis. So, so they did some pathway analysis using GSEA and other things on the genes that they found to be differentially methylated, and they didn't find anything. One of the reasons, was statistical reason with the distribution of data. We did the same type of pathway association study that I showed for the autism project. Instead, looking for association of methylation, differentially methylated genes in A versus B. Yes?
But A versus B could just be explained by the age difference, because there's a clear difference in methylation over time just by age.
Okay, so that's, yeah, so the comment is age, the age difference of these patients could explain the methylation pattern that's different. So that's an excellent point. I haven't considered that. I'll tell you more, just a little bit more about what happens, but that's an interesting point. In any case, it does seem to, it might be prognostically useful and practically useful. So that might be an explanation that's for this pattern, but it's still useful for them to know that that's a molecular signature that differentiates them.
Look at the mutual information between the age and then the CpG methylation pattern.
Yeah,
And
well,
if it's one, then you know
yeah, so it might be the case that it's age-related. We did this pathway association analysis. We used a large database of gene lists that we've collected from gene ontology and many pathway databases, and also the MSigDB, the molecular signatures database from the GSEA team at the Broad. And we found, interestingly, that there was really only one type of pathway that was enriched, and it was really, really super enriched. And it was all related to polycomb repressor 2 complex and targets of parts of that complex. Basically, out of more than 15,000 pathways that we searched, it was the only one that had any signal. And so it seems that the genes that were differentially methylated seem to be they probably methylated by PRC2, and that seems to be active in the A sample versus the A type versus the B type. And the interesting thing is that a experimental drug is available for repressing the methyltransferase, or for inhibiting the methyltransferase activity of one of the subunits. And they tried that drug on, and they got sort of an advanced version of that drug that's more potent, and they tried it on cell lines primary cell lines that they extracted from the samples in their lab, and it successfully killed all the cell lines specifically. The interesting thing about that, is that there's no known targeted therapy for this disease, so there's just surgery and radiation treatment. So because it would be the first potential targeted therapy, they can just try it right away in clinical trials. So that's the best result that we've had in terms of trying to gain mechanistic understanding of some genomics data sets. So we're presented with all this data, gene expression, whole genome sequencing, methylation, and then we compare it to known pathway information, and we find this really strong signal. And then they were able to find a drug that seems to kill the cell lines that they were looking at least. So that's a great example of, it's our best example of going from genomics to potential mechanism, and then possible translation. The other interesting thing, purely from a computational side of things, is that we don't usually see such a strong signal of like one or two, one gene set that comes up really strongly. And because we only got this one gene set, and it made the difference between the link to the PRC2 complex or not, we were actually able to find out where this gene set came from. And one of the curators for the GSEA MSigDB database in Jill Mesrov's lab actually read a paper, 2005 paper, and typed in the gene lists and put it in the database. And had he not done that, we never would have seen this connection. So it's also a really interesting, it's really good motivation for us to try and collect all of the mechanistic details that we can about the cell and put them in these databases so that you can search them. Because every now and then, one of them is going to be the key piece that you need to make a to make a link. So that's good motivation for us. Just some perspective, general perspective about pathway analysis. People we're interested, as I mentioned, in getting a mechanistic understanding of the system. That might be you might be interested in just understanding the biology, or you might be interested in perturbing the biology with finding a treatment for a disease. Most of the pathway analysis these days is really focused on gene set, looking at gene sets. The pathways are reduced to gene sets. So we know, as biologists, we know that pathways, there's a rich amount of information and detail in pathways. You can have quite a lot of detail about which protein is phosphorylating another protein and what that does. But the issue is that if you're working with genomics data, you want to say something about any gene that comes along. If you work at a very high level of detail, you don't have a lot of genome coverage. So there's this trade-off between how much genome coverage you have and the detail of the pathway information. So gene sets right now provide the most annotation for the most genes, and so they are most useful for genomics data, and that's why they're most popular. You can also have connections between genes, or you can know that one gene regulates another gene, or as I said, you might have biochemical detail about the pathways. You might know that a kinase phosphorylates a target. You might even have kinetic constants available that help you understand things in really a lot of detail. But as I said, this trade-off kind of makes it more difficult for us to use highly detailed data. But actually, interestingly, a lot of this data is becoming more and more available in databases. The Reactome database, I think, covers almost 10,000 proteins now with very detailed biochemical representations. And so this data is actually increasing in coverage. And future methods will really be trying to use more and more of this data to help gain a more detailed mechanistic understanding of the genomics data sets. So we can definitely do a lot with gene sets, but in the future, just perspective, people will be incorporating more and more of this detailed information to get more detailed information. So you might know eventually that there's a particular phosphorylation site in a protein that is important for the pathway activity that you see, and that's what's the key thing. And that would be really great if we could do that in the future. Okay, so there's a, do you guys want to, we have 30 minutes until the next break. Does anyone want to take a break now for a few minutes? Anyone want to take a break, or should I continue for 30 minutes? Continue, okay. So two more topics. Just a quick topic just to tell you about some of the technology that we're involved in building very collaboratively with many other labs to help collect information about cellular mechanisms and put them in databases. Right now, it's actually quite difficult to get all of the information that you'd like to get about pathways, for instance. And we maintain a website called pathguide.org that keeps track of all the pathway databases that have been published, all the databases that have some information about mechanistic details about the cell. And I have a summer student updating it this summer, and she's added like 150 of them. So now we have more than 500 databases that have some information about protein interactions or genetic interactions or transcription factors and gene regulatory networks, protein compound interactions, and there's a huge amount of information out there. So it's almost impossible. It would be impossible for you to, say I want all the pathway information about human system that I can find. So we're trying to, again in collaboration with many people, we're trying to develop some technology to help solve that problem. One of them is the BioPAX format. It's the Biological Pathway Exchange Language. It's a standard format that captures pathway information, metabolic pathways, signaling pathways, protein interactions, and gene regulation that has been developed as a community effort by many pathway databases. And the idea is that all these pathway databases have their own way of representing pathways. If they could all represent their data in one format, it would be much easier to collect data from multiple sources. And so that's the idea of BioPAX. BioPAX can represent all the sort of typical pathway, biochemical pathway level detail that you might expect, right down to phosphorylation sites and states of proteins and complexes. In collaboration with Chris Sander at Sloan Kettering Cancer Center, we've been developing Pathway Commons, which is meant to be a public library of pathway information. The idea is we'd like to make a one-stop shop for pathway information. It's available at pathwaycommons.org. Right now it has 9 databases in it. We're working on a second generation that will work with the latest version of BioPAX, and we'll be working on collecting as many pathway databases as we can, putting them in that one place. So that over time will become more and more useful. As I mentioned yesterday to people in the course, we have, and earlier, we have Cytoscape that we collaboratively develop again, mostly led by Trey Eidecker at University of California, San Diego, and started at the Institute for Systems Biology in Seattle as a network visualization and analysis software that's free. And then finally, putting a lot of these technologies together, my colleague Quaid Morris in Toronto and I have put together a system called Gene Mania. Gene Mania is trying to be like a Google search engine for gene function. So it does 3 things. If you have a gene that you don't know what the function is of that gene, you can type it into Gene Mania, and it will show you what genes connect to it, and that might give you some clue about the function of that gene. The second question that it answers is try to find genes that are related to a set of genes that I'm interested in. So if you have a set of genes that are somehow functionally related, and maybe they're part of a pathway that you're interested in, or they're associated with a disease that you're interested in, then you can type those into Gene Mania, and Gene Mania will recommend additional genes that are similar to that set. And it will look for genes that are highly connected. In that sense, it's very similar to the Amazon book recommending system or Netflix, where it recommends books based on what other people have liked. In this case, it recommends genes based on the genes that you give it, based on the connections. There's connections from many different sources: physical interactions, genetic interactions, predicted interactions, pathways, co-expression, protein sequence-related connections, and you can upload your own network information as well. So you could upload a weighted gene correlation network as one of the networks, and it would search that as well. So if I have time later, I can show it to you in more detail, but you can try it out at genemania.org. Finally, just finally with this section, I want to just make a note of the National Resource for Network Biology. So a number of labs that are involved in Cytoscape development got funding, NIH P41 research resource funding to develop a national resource for network biology. And Cytoscape is partially funded by this, but it's also funding social network analysis and other things. Generally, the goal is to promote the use of network analysis in biology and collaborate and train as many people as possible. So there are some training and outreach resources on this website, nrmb.org. Okay, so I'm going to acknowledge, I actually should have done this earlier, acknowledge Enrichment Map people, and then I'm going to move on to another topic that's quite different, but somewhat related. So I mentioned Ruth Isserlin, Daniela Merico, Oliver, and Andrew Emili, who worked on the original enrichment map. Ruth is the programmer who built the plugin, and I mentioned these Michael Taylor, Steve Mack, and Scott Zeiderdine are postdocs, students in Scott's in my lab and Steve is in Michael Taylor's lab, who did the work on the Penamoma. And just as a brief interlude before I switch topics, Ruth was, when she wrote the enrichment map software, she was really getting into it. And so when she presented it at a lab meeting, she baked an enrichment map cookie. And so I can tell you that enrichment maps are not only useful, but they're also quite tasty. Okay, so that's just, I'll now transition to the second topic. See how much time I have to go over this, but this is another way of thinking about using mechanistic information, biochemical mechanism to gain more insight into genomics data sets. In this case, the genomic data set that we're thinking about is mutation data sets as mapped by the Cancer Genome Atlas Project, or the ICGC, the International Cancer Genome Consortium, which, as many of you know, might know, they're sequencing thousands of tumor samples and exome and whole genome sequencing and identifying mutation profiles in all these tumors. So I'll skip over that. So these are, these are papers relating to all these cancer genomics projects. There's a huge amount of information coming out of these projects. And one of the central challenges is to try and identify which genes are mutated and causing cancer and called drivers versus which ones are just mutated because cancer has a lot of mutations and there's problems with DNA damage. And those are, people typically call them passenger mutations. So the main way that people distinguish the drivers versus passengers is by looking at mutation frequency. So if a gene is mutated over and over again in different independent samples, it's very unlikely that that is due to chance, and you would expect that that has something to do with cancer tumor progression and development. And people have seen certain genes mutated a lot more often than others. If you just look at, we looked at 800 cancer samples from 8 types, and we found that, and people have seen this, that P53 and KRAS, EGFR, PTEN are mutated, a huge amount of times. But most of the mutations are actually captured in a very long tail of lower mutation frequency, but a lot of known cancer genes are found in this tail. So there's a challenge of kind of trying to find more information in this tail. So the idea that and this is work from a postdoc in my lab, Yuri Riemann, who we had an idea to use additional information about biochemical pathways, in this case protein phosphorylation information, to try and get a better understanding of trying to find more cancer drivers. And I'll get, there's another network analysis part later, but this is more sequence analysis. The idea is that we're looking for mutations that are recurrently found in a specific region that's phosphorylated or in a kinase domain. So instead of looking at the whole protein, you look at an individual region in the protein. And if you see, for instance, you might expect that a given protein might be mutated a given amount of times based on which tumor it's mutated in. But if all of those mutations are really focused in one particular area, then that's significant. So we looked at 800 samples from 8 different tumors, and we found quite a few genes that were mutated in 136 genes that were significantly mutated at specific phosphorylation sites. And we call this active driver. And this plot shows where those are found. So all the gray lines are known cancer gene, all the gray lines are genes that we predict are mutated more than you expect. And the red lines are known cancer genes. So some of the known cancer genes aren't mutated that often in 800 tumors. And we find we predict some known cancer genes that are in this region here, which sort of validates the approach. Here's an example in beta-catenin where you have mutations affecting, a lot of mutations seem to be affecting phosphorylation sites. And we also know kinases that are responsible, at least some kinases that are responsible for phosphorylating these sites. And so we can look at that, and that might give us a hypothesis that this mutation is not just related to cancer, but it's actually potentially removing a phosphorylation site that might be an important signaling switch in the protein, and that might be, the mechanistic reason for development of increased growth or increased resistance to apoptosis or something related to cancer. Okay, so we also did a pathway enrichment analysis. I won't go into detail on it. We developed a new network kind of search method that tries to find regions in a network. In this case, we had a network of kinase substrate relationships, a few thousand of them downloaded from the Phosphocyte Plus database. And we were looking for regions of this network that were both mutated, significantly mutated, and also correlated with clinical outcome. In this case, we looked at the only major clinical outcome that we had available at the time, which was from the ovarian cancer genomics project, and we looked at survival. So the idea is that it's not really a association test. It's more like a search algorithm. So one of the problems we had with these methods is that even though we found lots of predicted cancer drivers, and we found pathways that were enriched in mutations, again, in these phosphorylation regions, we didn't find any correlation with clinical parameters. So we were kind of looking for some correlation, some signal that correlates with outcome, for instance. And so that's the reason why we developed this method. We want to just look at the network and search that network to see if there's any region in the network that's correlated. So the way this works is we take a gene of interest and we look at its neighborhood up to 2 hops, and we include parts of the neighborhood, and we look at the mutations in the proteins in that neighborhood, and we do we run some statistical test that measures correlation with a clinical variable. So for survival, we use Cox proportional hazard or log rank statistic. For a categorical variable, we use Fisher's exact test. And so we look in a neighborhood and we say, okay, are the genes in this neighborhood correlated with the clinical variable? If they are, we keep that part of the neighborhood. If they're not, we throw it away and we look at another part of the neighborhood. And so we kind of build up a little neighborhood around a gene, focusing on the genes that are correlated, the mutations that are correlated with the outcome. This is very biased. It's looking for things that are correlated, but it's limited in a couple of ways. Well, it's limited in that it can only follow short little neighborhoods around individual genes. But you could have a lot of correlation by chance, and so we do a lot of permutation testing to check to see if a module is likely to occur by chance by rearranging all the mutations in the network and running the algorithm 10,000 times, and so we can get a p-value for how likely a particular signal is to occur. In the end, we found one module that was correlated with outcome. All of these genes around ABL1, ABL1 wasn't mutated itself, but a lot of its neighbors were. If they were mutated, they were correlated with good survival versus the population, and similarly as other clinical variables that were related were correlated in ways that you would expect. Again, this is not that many patients covered, so there's only 8 patients out of 800. We're currently working on a data set that has 3,500 samples, and so hopefully we'll get better statistics. So I think this is not really a method to say that this is now a signature that you can use. It's more a search method that you can use, again, to generate a hypothesis. This is a region of the network that seems to be correlated. Maybe that's interesting to follow up on. Okay, so I mentioned there's cancer genomics projects are growing rapidly, and the latest generation from the TCGA is the pan-cancer project, where they've collected up to, I don't know, I think if you count all of the different types of data, it's like 4,000 or 5,000 samples, but they have at least almost 3,200 that have exome sequencing and from 12 cancer types. And so we've run our analysis again on that data. We also added a little bit of additional, one additional aspect of the analysis, which is looking at how the kinases recognize sequence motifs in the protein and seeing if not just the sequence, the mutation affects the phosphorylation site, but if it's predicted to rewire the network in some way. So either, well, obviously if it removes the phosphorylation site, if a mutation kills the phosphorylation site, kills the central phosphorylatable tyrosine, serine, or threonine amino acid, then it's not going to be possible to phosphorylate it. But if there's a mutation in the flanking region, that could change the kinase that's recognizing that region. And we can try to predict which kinase it might be changing it to by searching all known kinase specificities. Kinases recognize sequence motifs around the phosphorylated residue. And so here's an example where, again, in beta-catenin, sort of kind of our control, we have a mutation in the phosphorylated position, and we also have a bunch of mutations from different samples in nearby to the phosphorylation position. And if you look at different kinases and what sequences they recognize, those mutations are removing a important position that if this kinase recognizes this position, it would now not be able to recognize it. So this sort of expands the amount of hypotheses that we can make, and also it expands the detail of the hypotheses that we can make. We can say that we might be able to say or predict that a particular mutation is causing a particular kinase not to be able to phosphorylate that protein, and that might be important. Or we might be able to say that a mutation causes a new site for a kinase that was never able to phosphorylate that site before, and that might be important. So that's another topic, but just another example of how to combine genomic data, which gives us kind of static state, snapshots of the system, and cell mechanism, biochemical mechanism information, and try to get some mechanistic insight. So it's not just pathway analysis. This is maybe a more detailed type of pathway analysis. And I think this is an example of the types of things that will happen more and more in the future as we get more and more of this detailed data, like phosphorylation sites and things like that. So this is more of a research topic. It's not. There's an R package actually for active driver, which is freely available. The paper's published in January. And so if people are actually interested in doing this with their sites, it doesn't have to be phosphorylation sites. It can just be any site in a protein. If you have sites of interest in proteins, you can assess whether mutations are, a given set of mutations is affecting those sites more or less than you would expect. Okay, so again, this is work by Yuri Raymond, a postdoc in my lab, and Omar Wagi, who is a master's student. Okay, so that's it. Any questions? Okay, so that's it. Thank you very much for your attention.